So phosphatidylglycerol is condensed with CDP-diacylglycerol by cardiolipin synthase, and a now classic paper by Michael Schlamy back in the early 90s. He demonstrated that this actually, the active site of cardiolipin synthase in, I believe, rat liver mitosis, was on the matrix side of the inner mitochondrial membrane. So newly synthesized cardiolipin is made, and all the enzymes that precede it, really the active site in terms of the membrane is on the matrix side of the leaflet. Now, we all know that there's cardiolipin remodeling, and in mammals, there's at least 3 and possibly even more remodeling pathways, but they're original. The evolutionarily conserved remodeling pathway is the one that's mediated by tophasin. So you have immature cardiolipin, which is characterized by a random attachment of saturated acyl chains. You have some type of lipase or phospholipase that comes in and cleaves off an acyl chain, generating monolysocardiolipin. And then tophasin transacylates, uses other lipids as the source of acyl chain to convert monolysocardiolipin back to cardiolipin. And through a series of these cycles, you're able to go from this form to what you normally see at steady state. Now, topologically, this, from work that we did back in the 2006, we showed that the yeast tophasin actually associates with both the outer membrane and the inner membrane, but faces the inner membrane space. And it does so through a membrane anchor that doesn't seem deep enough to necessarily gain access to the opposite leaflet, although that's, I think, still just speculative. Now, this localization has been confirmed subsequently by Klaus Fahner in collaboration with Miriam Greenberg. So there's an issue here. You have cardiolipin being made on this side, but the enzyme that's going to finish the remodeling in yeast is on the opposite leaflet. And I think it's easy to appreciate that flipping a dimeric lipid with 2 head groups and 4 acyl chains is not going to be necessarily a simple process. And so a couple years ago, in yeast, the enzyme that initiates the remodeling cascade was identified, cardiolipin deacylase, and I'll talk about that for the rest of the talk primarily. And it's a big black box here because it was not localized within mitochondria. And so you can envision that perhaps this lipase, its activity is coupled to movement across the lipid bilayer, or perhaps they're separate processes, and that would imply that there needs to be other activities that are then going to move this lipid back and forth across the leaflet of the bilayer. So as we know, if you delete tophasin in any model system that has been tested to date, what you see is a lack of the mature form of cardiolipin, an increase in the intermediate monolysocardiolipin, and the acyl chain composition of the remaining cardiolipin seems to be abnormal. So a little more about cardiolipin-specific deacylase. It's identified in yeast. It co-localizes with mitochondria by fluorescence. This is with a GFP-tagged form. It co-fractionates with mitochondria using a GST-tagged form. Both of these constructs use tags placed on the N-terminus, which is an odd choice, but they did work. And then it was noted that the deletion strain gave you increased abundance of 16-0 containing cardiolipin at the expense of 18-1 and 16-1. Finally, they nicely showed that you can this enzyme CLD1P could deacylate both cardiolipin and PCs, which is notable because tophasin itself is known to be able to remodel multiple different lipid species. So these are 2 enzymes implicated in one pathway that do have the capacity to influence more than just cardiolipin. So just to look at the next 2 slides, will have the only published data of the talk, and they're not mine. So down there, it's a little cut off. Baranek and all published, and this is their lipid biochemistry epistasis analysis. And so what they did is they're looking at the lipids in their different yeast strains. And here's their wild type strain, normal profile. You have this delta YGR 110W, which is CLD, looks like wild type by this analysis. TAS gives you the expected decrease in cardiolipin, and you see the hallmark monolysocardiolipin, and really the biochemical hallmark, as Fred's shown, is the ratio of these. And importantly, when they had a double deletion, they showed that this phenocopied the single CLD mutant. evidence that CLD functions upstream of tophasin in the same biosynthetic pathway. Now, it gets a little more complicated when they did a growth epistasis analysis. And so focusing down here, when you start to challenge yeast to grow on ethanol, or glycerol, or lactic acid, which require mitochondrial activity, you see that tophasin maybe has a slight defect at elevated temperature on glycerol ethanol. There's no obvious phenotype for the single CLD1 mutant. Oddly, the combined mutant had an additive effect. And the same story for lactic acid. And in terms of genetics, this made no sense with them functioning in a linear pathway. It actually argued that they functioned in distinct pathways. So there was a little bit of a discrepancy that didn't quite make sense to me when I read this. And so our first inroads into kind of trying to ask a cell biological question about CLD1 was to look at this epistasis analysis. And so we generated a panel of mutants that were either single or double mutants in cardiolipin synthase, cardiolipin deacylase, and tophasin. So we added cardiolipin synthase into this epistatic analysis. And you can see wild type gives you the normal profile, no cardiolipin synthase. You have phosphatidylglycerol accumulating. CLD, consistent with Baranek et al., looks like wild type, delta TAZ, as you'd expect. You have the appearance of monolysocardiolipin. If you don't have cardiolipin synthase, regardless of whether you're missing these other enzymes, you have phosphatidylglycerol, so cardiolipin synthase is the first enzyme in the pathway. And if you have the combined double mutants, exactly what Baranek et al. Published, this phenocopies the single CLD mutant, which phenocopies the wild type in this analysis. So we got a different result when Matt did growth analyses with our standard wild type strain, which is GA741A. Focusing just on the ethanol, if you grow them at 30 degrees, you see a growth defect in the absence of cardiolipin synthase, in the absence of the deacylase, so you don't initiate remodeling, it grows like wild type. TAZ has a growth defect. Anything lacking cardiolipin synthase has a growth defect, and the double mutant grows like the single mutant, which grows like wild type. That's our strain background. So this is consistent with it functioning in a linear pathway. Since we saw a different result than what was published, we wanted to repeat this analysis in a second background strain, W303, which is a lot more of a standard laboratory strain than GA74. And the story holds true. If we grow on ethanol glycerol at 37 degrees, you see the same profile. Single mutant of this lipase grows as well as wild type, and the double mutant grows as well as a single mutant, which grows as well as wild type. So really this indicates that cardiolipin deacylase, this cardiolipin-specific deacylase, acts upstream of tophasin and downstream of CRD, downstream of cardiolipin synthase. So it's right smack dab in the middle of this remodeling pathway. So if you're not initiating remodeling, presumably the cardiolipin that you accumulate will have an abnormal composition of acyl chains. So together with Jinlin Han, he did some shotgun lipidomics analyses for us. And this is complicated because anything with an omics in its name is. So we're going to provide some arrows in red. These red arrows highlight the major molecular species of cardiolipin in our wild type yeast strain. You see they're characterized by a content of 18.1 and 16.1 in variable proportions. But by and large, I think 90% of the cardiolipin that you detect at steady state and wild type are one of these four. Now, if you compare that to a strain lacking the cardiolipin deacylase, you see a drop in what you normally saw for wild type, and you see an increased abundance of other forms of cardiolipin. So you see a molecular diversification of the cardiolipin species when you don't initiate remodeling. And if you just will quickly note, in green is the double TAZ CLD mutant, and it basically looks like the yellow bars in the histogram. So then what Matt did, he's a clever grad student, he decided to analyze this data in, I think, kind of an easy way to digest. And so what he did is he looked at the total amount of cardiolipin, and then he characterized whether the cardiolipin had no saturated acyl chains, one saturated acyl chain, or two saturated acyl chains. And you can see in wild-type yeast, by and large, 90-95% of the cardiolipin molecules have no saturated acyl chains. Now, if you take either the single cardiolipin deacylase mutant or the double TAZ CLD1 mutant, the profile is the same, and you see a lot less of cardiolipin molecules with 0 saturated chains, a huge amount of cardiolipin with 1 saturated acyl chain, and a good proportion of cardiolipin with 2 acyl chains. Now, what's interesting, if you look at this for tafazin, you see that what remains, and so this asterisk is here to show me this is the way this is presented is whatever's there is 100%. It's noted when you look at the actual amounts of cardiolipin in the TAZ mutant, it's about 25 to 30% less than in wild type or CLD. So this is a smaller absolute amount, but the proportion is what we're looking at. And if you look at the proportion, it actually looks somewhat decent. I mean, there's quite a bit of cardiolipin with 0 saturated acyl chains, a little bit with 1, and a small amount with 2 saturated acyl chains. And actually, if you compare this value in terms of absolute amounts with this value right here, you actually can get the quantitative amount of cardiolipin that likely comes through the biosynthetic pathway already in a mature state. So since Matt had kind of worked out some of this stuff, he was interested in mapping the catalytic residues of this enzyme that initiates the remodeling pathway. And so the original description by Baranek et al., they highlighted that this enzyme had an alpha-beta hydrolase domain, and they highlighted 2 potential motifs that had similarity to lipase and or acyl transferase motifs, as well as an acyl transferase motif. And you can't really see it here, but they showed that there was a serine right here, as well as a potential histidine and aspartic acid that might be catalytically important. Now, Matt took a class in grad school that dealt with structure and modeling and stuff. This is one of these things that I need to take this class because this is pretty cool. And what he did is he then tried to build a homology model of this enzyme, and he used as his kind of template this bacterial hydrolase CUM-D, which was a weak homology model, but still was useful. And so he wanted to see if these original residues kind of seemed to kind of come together in a common catalytic motif, and also potentially identify other candidate amino acids that we can mutate and see if they're involved. And in fact, he found a cluster of aspartic acid residues that were in a separate region of the protein that were in this region and were therefore of potential catalytic residues. And so what our strategy was to take advantage of the simple lipid profile by TLC. So if you have a yeast strain that lacks tafazin and this cardiolipin deacylase, you have a wild-type looking profile, but we know by mass spec that acyl chain composition is not normal. Now, if you have an active cardiolipin deacylase, but you have an inactive tafazin, you have elevated monolysic cardiolipin. All right? So then you can go back here, where you have this situation, make your mutant constructs, transform them into a yeast host that lacks both of these enzymes endogenously, and ask, does it work? And if it works, you're going to start generating monolysic cardiolipin. If it doesn't, you won't. Okay? Simple. Trust me. All right, so this is what it looks like. So the background strain of interest is a strain that cannot remodel cardiolipin and cannot initiate it. So if you transform this with the empty vector, and this is looking at radioactive lipids, you see only cardiolipin. If you transform that strain back with a wild-type form of this enzyme, you see monolysic cardiolipin. So this means active. This means no activity. So if you mutate this serine and this histidine, you get no activity. These two, this other histidine, which is right next door, wild-type activity. This aspartic acid, which was originally thought to be involved in the catalytic triad, you get activity. So it's not involved. One of the homology model identified aspartic acids, no activity. And importantly, if you look at the steady state levels, all this isn't the prettiest blot. CLD is expressed roughly as similar across the board. So expression isn't why these are inactive. It's that they are inactive. He's also gone on to show by measures of assembly, blue native page, that these all assemble normally, which kind of pinpoints that these are actually catalytic motifs and not grossly disrupting the folding of this enzyme. So the catalytic triad of cardiolipin-specific deacylase is highlighted in the darker colors. It's consisting of serine 230, histidine 424, and aspartic acid 392. So let's get to what is actually the topic of the talk is where does this fit in this topological pathway? And so as I showed you just a couple slides ago, we made antibodies against CLD1P, and Matt confirmed what had been shown with the epitope tagged construct, that endogenous bona fide CLD1 co-fractionates with mitochondria, as shown here by its presence in the enrichment in mitochondria by showing a blot for QCR6, which is a subunit of complex 3. Next, where is it in mitochondria? And so what Matt took advantage of is the fact that digitonin can differentially solubilize different compartments of mitochondria. So as you add low amounts of digitonin, the first compartment to be solubilized are proteins that are soluble constituents of the intermembrane space. So if you then add low amounts of digitonin, separate that into a pellet soup, soluble IMS stuff is released. As you start to add a little bit more digitonin, you next release the matrix, then the outer membrane because it's less protein dense than the inner membrane, and then finally you start to release the inner membrane. Okay, and so what he did is he tracked 2 proteins per compartment, combined the data, and came up with this histogram. So you can see his soluble IMS comes out really easy. This is the percent of total in the supernate, and this is the amount of digitonin. Next out is matrix, next out is outer membrane. In purple is the inner membrane. In blue, CLD looks most like the inner membrane. So this is on the inner membrane. But this doesn't give you any sidedness. We don't know which side of the membrane it's on. And so what he did is a proteinase K experiment. And so in this experiment, you basically take mitochondria and do different things to them in the presence of proteinase K. The first condition is you just take mitochondria that you've isolated, add proteinase K, and you can see here that an outer membrane protein with a large domain on the cytosol is degraded, whereas a protein anchored to the inner membrane with a large domain facing the inner membrane space is not, nor is a protein in the matrix. Next, you can take mitochondria, put them in hypoosmotic conditions, causes them to swell, physically ruptures the outer membrane. Now protease has access underneath the outer membrane, but still can't get to the matrix. So the outer membrane is still degraded, but now the inner membrane marker is degraded. Matrix protected. Finally, you can add detergent and all compartments become accessible to protease. And you can see if you just have the right controls, you compare the profiles, CLD is on the matrix side of the inner membrane. Next, we wanted to get at the question of whether this lipase activity could be coupled to movement of lipids by getting an idea of the nature of its membrane association. So Matt devised a very complicated schema that actually is quite informative, but it's a little bit of a challenge to present. So we're going to walk through it schematically first. So here you're going to take swollen mitochondria, so you've ruptured the outer membrane, and you're going to add different amounts of salt. Salt is known to disrupt electrostatic interactions. So the classic protein that we'll track in this assay is cytochrome C, which is known to be peripherally attached to the inner membrane through electrostatic interactions. So you take those mitoplasts, either don't add salt or add increasing amounts of salt, spin it in the soup, or electrostatically attached proteins or soluble proteins stuck to the pellet or everything else. Next, you can take these mitoplasts, again, don't add salt or add increasing amounts of salt, sonicate, so you're going to add bursts of sound and basically rupture the membranes into little bits, so now salt will have access to all sides of the membranes, of all membranes. And then do an ultra-speed spin, and you separate that into a supernate and pellet. From 20 millimolar to 1 molar. So he did a fairly good range. And so we'll first focus down here, because this is our peripheral intermembrane control. So you have your IMS fraction, which is the mitoplast with or without salt. And you can see for cytochrome C, it's not released until you start to titrate up the salt. I think right here is where you start to get quantitative release, and so you don't see it in any other fractions. So right off the bat, if you look at the profile of cytochrome C, which is exactly what you'd expect based on everything that we've done previously and that has been published, and compare it to that of CLD, you never see CLD released into the IMS under any amount of salt. So whatever salt may or may not do, it's not doing it to CLD on the IMS side of the inner membrane. Now, if you look at the P and S, which are the sonicated stuff with and without salt, you see that without any salt added, it's in the pellet, it's in the pellet, it's in the pellet, it starts to be in the soup, starts to be in the soup, starts to be in the soup, and now when you add basically Thor's hammer of salt, I mean, that's a ton of salt in this assay, you start to get release of CLD1P. And what this implies, it suggests, is that electrostatic interactions are really important for this enzyme's association with the intermitochondrial membrane, and it's also important on the matrix side. This is all happening on the matrix side. And so when you put this into the cartoon that we've been building, what this shows is that it's the remodeling pathway, and at least in yeast, is also being initiated on the matrix side of the intermitochondrial membrane, which leaves us wondering, how in the world is monolysic cardiolipin getting access or flipping to the opposite side where tafazin can then remodel it? And so we're speculating that there needs to be some activity, a scramblase, a flopase, a flipase. The flopase is supposed to have 2 Ps, I don't even know. That is perhaps has specificity for monolysic cardiolipin that's mediating this activity in yeast, and it's obviously something we're very interested in finding out. So this is my group. Most of the studies I've been talking about were performed by a really talented graduate student, Matt Bale. These other guys try to keep them in check, but they also do call him Little Boss, which is good because it implies that he's little and he's actually a lot bigger than me. Some of the collaborators this work started in Carla's lab, Jinlin Han for shotgun lipidomics, who's now down at the Sanford in Burnham, and Mike McCaffrey, who has a long-standing collaboration with us on EM. Funding by the National Heart, Lung, and Blood Institute, and also the Barth Syndrome Foundation. Done. Thank you.
Beautiful work. So how you, let me ask you an obvious question, then more difficult one. How you reconcile the differences between your complementation experiment versus the lipidomics data? Because obviously when you knock out both CRD and CLD, you have a wild-type phenotype. But the cardiolipin profile are very different.
No, when we knock out CLD and TAS.
TAS, sorry, yeah CLD and TAS, yes.
The lipid profile is different between.
I mean, the cardiolipin profile is very different from wild type, but their behavior is completely normal.
Yes.
As long as we can see based on the morphology.
Yeah. That's true. You're absolutely right. So I mean, in Baranek et al.'s paper, they use this as evidence to support the notion that remodeled cardiolipin is important for mitochondrial function. But genetically, it just didn't make sense, their data. And our data is actually a lot more consistent with what you've shown in flies, where, if I forget exactly what you did, but I know if you added BEL or something, right? I mean, and you saw that there was some, I forget the details, but I know it's consistent with what you guys saw. So, it begs the question of how important the molecular forms are, right?
Then more difficult question is, what happened?
That's a pretty difficult question, actually. I mean, we can get philosophical, but
so this is the Barth Syndrome Foundation symposium. What's the relevance to the human tafazin? If you could knock out the human version of a CLD1, would you rescue the defect?
Well, I mean, so in mammals, I think there's likely to, and in other eukaryotes, there's likely to be more than one lipase that's involved in this pathway. I think that's probable. I mean, IPLA2 gamma seems to be involved, but I still think there's some missing pieces in how directly it's involved in what's the proportion, right? I think a lot of this needs to be built up into some of the topological issues that we've raised in yeast need to be verified in mammals to see if they're conserved or not. And these are works in progress. But if one was to assume that the topology of this pathway of remodeling is conserved, which again it may or may not be, this does offer some targets, right? So you can target not only the lipase, but also perhaps the molecule that's mediating the flipping, right? Perhaps, or other things. So it does offer some potential therapeutic targets, assuming these are conserved processes. But that's an assumption that I'm not actually willing to make.
I was wondering whether the H4XD mean anything in tafficin. In your case, when you mutate it, does it complement or not? In your case. So
one of the Barth syndrome mutants, I chose not to present the rest of the characterization that we've done on the panel of Barth syndrome mutant taphasins in yeast. But one of them, actually BS1, according to my silly nomenclature when I started this, is the supposed catalytic histamine. And I can tell you that it's expressed at wild-type levels. It assembles completely normally. Its lipid profile at steady state's like a null. And thanks to Michael, I was able to establish transasolase assays in our lab, and it's completely dead. So it smells like a catalytic mutant. We haven't mutated the aspartic acid, but it makes sense too. And get at another thing, the fact that this HXX whatever D motif here, the aspartic acid in CLD1 was not involved in catalysis, right? And so whether that tells you that it's more of a lipase than you get into different questions.
We got time for one more.
Over here.
Oh, excellent. Steve, just to follow up on and we've discussed this, the difference between maybe what you end up with in terms of cardiolipin composition and what the process of remodeling may result in terms of the cell function or morphology. And so I think this story adds another layer to that in terms of all the movement that may be going on in this membrane to get this process done. And at the end, you may have a molecule that has some kind of compositional uniformity. But you've also flipped and flopped and turned and who knows what else. And so my question at this point is, especially since you mentioned collaborating with Mike McCafferty on EM, have you looked at the membrane morphology of these yeast strains and seen any kind of changes in, like, I don't know if they have cristae or if the membranes are folded, or any kind of differences in any of these mutants that would suggest that this process is controlling the structure of the membrane or its formation?
Those are private conversations. Yes, we have. His picture is there. It's still preliminary, is what I'd say. We do see differences, and I don't want to go into them because I want to confirm what we're seeing we've only looked at them in one genetic background in one set of samples, and I want to confirm it in a second set of samples and a second genetic background before we get excited. And I don't want to, it makes no sense to get excited for anyone if it's something I don't believe in yet. But perhaps there's some structural differences.
Fred, you had a real quickie?
Yeah, I just wanted to comment that the strains, the W303 and the GA74, had very different phenotype with relation to different mutants. So wouldn't that be consistent to look for modifiers of the phenotype you're looking at?
Yeah, Matt, when I pitched this little project to him to kind of make sure that he'd graduate, because it was going to be simple. We had antibodies here, just do it. It started out with growth studies, and and I'm sure as Miriam can talk about, different strains give you different behaviors, although the trend was the same, right? So if you compared ethanol for the two media as an example, W303 probably grew fine, but GA74 didn't. So you had to kind of find the right conditions that teased out the phenotype between these strains, and I imagine that was an important lesson for us. But it's also why it was important to do it, and again, why I don't want to talk about the morphology until we've confirmed it in a second background.
Thanks, Steve.
Mm-hmm.
So the second speaker today is Miriam Greenberg.